Very good evening, everybody. I am going to discuss on the topic accessory respiratory organs of fish. And this topic is mentioned in this category. That's a Joule 2011 Codex, the paper. All of you know that the fishes are the aquatic and they possesses gills for branchial mode of respiration. However, in many fishes, either due to poorly developed branchial respiration or due to environmental pressure, additional respiratory structure have been developed. Such extrabranchial organs that supplement gills in respiration are collectively referred as accessory respiratory organs. The development of accessory respiratory organs is found mostly in freshwater fishes of tropical region and very rare in marine fishes because marine water contain huge amount of oxygen. So usually, the marine fishes has no accessory respiratory organ, but few exceptions are present, such as Scylochylium indicum and Chyloscylium griseum. And accessory respiratory organs that are present in the teleos, such as external gills that is found in Dibnon's larvae. Integument also serve as accessory respiratory organ that is found in Amphibians cuchia, Anguilla, and Periophthalmus. Oral papillae also serve as accessory respiratory organ in case of Electrophorus. Buccopharyngeal epithelium serve as accessory respiratory organ in case of Monopteras. Intestine, basically posterior intestine, also serve as accessory respiratory organ in case of Lepidocephalectis gantia. Labyrinthine organs serve as accessory respiratory organ in case of Anabastes studenius and Trichogaster. Opercular chamber serves as accessory respiratory organ in case of Periophthalmus. Opercular lung serves as accessory respiratory organ in case of Clarias batrachus, Heteroponestis fossilis, Anabastes studenius, and Trichogaster. Ear bladder also serves as accessory respiratory organ in case of Notopteras, Polypteras, Amia, Lepidosiren, and pharyngeal lungs serves as accessory respiratory organ Channa amphibians. And all these fishes have gills, but what are the needed for accessory respiratory organ? Several reasons have been suggested: to meet the environmental oxygen deficiency, to compensate the degenerate gills, to maintain life during estivation period that is known as summer sleep, to counterbalance the internal oxygen deficiency, production of much carbon dioxide in water by their flora, fauna, and fishes themselves as well, causing formation of carbonic acid that is the aquatic toxicant, and air breathing may be obligatory in some fishes. To overcome this adverse situation, several types of accessory respiratory organs functionable in aquatic or aerial environment that have been evolved in fishes. The development of such structure is essentially adaptive in nature. Some accessory organs serve aquatic respiration, while others for aerial respiration. The accessory respiratory organs of the fishes, such as skin, in case of Anguilla, and in case of Amphibians, Cuchia, Mastasemblas, Pancalas, the skin is highly vascular and serves for exchange of gases. They live in oxygen deficient stagnant water and migrate one place to another through damp vegetation. During this period, the moist skin, which is supplied with blood, that serves as important organ for respiration. Then buccopharyngeal epithelium, such as in case of Amphibians cuchia, Boleophthalmus, Periophthalmus, the buccopharyngeal epithelium is supplied by large number of the capillaries. In case of the fish, buccal region and pharyngeal region are conjugated and they form buccopharyngeal region, and that's covered by a membrane. The membrane consists of large number of the capillaries and that's form vascular network which work as efficient respiratory sites. In case of muddy skipper, such as Periophthalmus and Boleophthalmus, live water and they spend most of the time skipping or walking about through dampy areas, particularly around the roots of mangrove trees in Sundarban region. Fresh air is gulped into the cavities and exchange of the respiratory gases occurs through the epithelium. Then next, pharyngeal diverticulum. In case of Channa species, it may be Channa gachua, Channa status, Channa punctatus, Channa marulius. The suprabranchial cavity. This is a suprabranchial cavity that are developed in the roof of the pharynx, and suprabranchial cavities possesses some alveoli. The first gill arch appears to have greatly flattened and covered by thin vascular respiratory membrane which becomes highly folded and studded with large number of the papillae or nodules that forming labyrinthine or dendritic organ. The respiratory surface extends anteriorly to the roof of the buccal cavity, and the surface of the tongue, the suprabronchial chamber, and the entire buccopharyngeal cavity serves for aerial respiration, and that is filled with air during the process. In case of amphibious cuchia, the air breathing organs are in the form of pair of sacs and that are located in the lateral sides of the head, and these are the diverticuli of the pharynx that are lined with vascular epithelium that's thrown into folds and ridges, and each sac is provided with inhalant aperture and exhalant aperture. The respiratory epithelium of the air sac consists of vascular areas in the form of islets bearing many rosetti or papillae. The thin epithelium having network of capillaries that covers with islets. In case of periophthalmus, a small shallow pharyngeal diverticuli lined with vascular epithelium is present on each side of the roof of the pharynx. Now, next, oral papillae. In electrophorus, the gills are degenerate. To compensate, the mucous membrane of the mouth that raised into the oral papillae, they are well vascularized and they help in aerial respiration. Alimentary canal. Uh, in some fishes such as Lepidocephalectis gantia and Misgarnas fossilis, the epithelial lining of the certain parts of the alimentary canal becomes vascular and modified to serve as a respiratory organ. The inhaled air is swallowed and forced back into the alimentary canal, and that is stored sometimes in the special part that is a posterior intestine. And after gases exchange, the gas is voided through the anus. And you see here the gut wall, especially this is the hind gut, this is a posterior regions, the area becomes thin due to a reduction of the muscularis layer, and here the sub mucosa is highly vascularized. So these blood vessels that are responsible for respiration. And air bladder that is also modified in some fishes and also serve as accessory respiratory organ. In case of Notopterus, they have a white pneumatic duct and act as accessory respiratory organ. The network of blood capillaries covered by a single layer of epithelium facilitates diffusion of gases between the blood and the air contained in the swim bladder. The dipnoid, uh, that is the air bladder, is most highly evolved, acting as a lung. It is vascular. It is a highly vascular and that contain alveoli-like structure. In Amia lepiosteus, the single air bladder is present, which opens dorsally into the pharynx, whereas in case of the Lepidosiren and Protopteras, the air bladder is bilobed, ventral, and opens ventrally into the pharynx. In some fishes, pelvic fins also serve as respiratory organ. In American lungfish, Lepidosiren, during breeding season, the pelvic fins of the male fish enlarge in size, become highly vascular, and forms filamentous outgrowth. This fin serves as accessory respiratory organ, supply oxygen to the eggs guarded by them. Then next, opercular chamber that is modified and that also serves as respiratory organ. In some species such as Symbranchnus marmoreti and Periophthalmus, the inhaled air is passed through the gill slits into the opercular chamber where it is stored for some times. The membrane lining the opercular chamber becomes thin and highly vascular to allow exchange of respiratory gases. Next category, opercular lungs or bronchial diverticuli. In air-breathing fishes such as Anabastes studenius, Trichogaster, Heteroponistes fossilis, Clarias batrachus, sac-like diverticuli develop from the dorsal surface of the opercular or bronchial chamber. These air chambers that is known as opercular lungs that lie above the gills and may contain specialized structure. They are known as arboration organ or dendriform organ or rosette-like structure to increase the respiratory surface area. The air-breathing fish, in case of the Anabastes studenius, the organ that consists of the spacious air chamber either side of the skull, lying between the first gill arch and the hyomandibular arch. This organ is labyrinthine organ that develops from the epibranchial segment of the first gill arch and consists of three concentrically arranged bony plates. The first plate is fused with epithelium lining the air chamber, while the second and third plates are much folded and highly complex, that's covered with vascular epithelium, which serves to increase the area for the absorption of oxygen. In case of the trichogaster fasciatus, the respiratory organs that consist of suprabronchial chamber, this is a suprabronchial chamber, and then labyrinthine organ, and also contains respiratory membrane. The suprabronchial chamber, it is situated just above the gill. You see here gill, just above the gill, and 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 either sides of the either sides, and in case of the Anabas testudineus, in comparison to Anabas testudineus, it is more simpler structure, and the labyrinthine organ that develops from the epibranchial part of the first gill arch, and it is and it is in the form of the spiral organ that possessing two leaf-like extension, and it is composed of loose connective tissue covered by a vascular epithelium. The respiratory membrane lining the air chamber and covering the labyrinthine organ that consists of vascular and non-vascular area, of which the vascular area that possesses a large number of the islets that containing parallel blood capillaries, and the islets are believed to be derived from the second lamellae of a typical gill filament. Then in case of Heteroponistes fossilis, the accessory respiratory organs that consist of expanded gill plates, air sac, and also respiratory membrane. A pair of simple sac-like structure that extends posteriorly from the suprabronchial chamber up to the middle of the caudal region. The air sacs are thin-walled, long tubular structure with highly vascular walls and embedded between the myotoms of the body. The air sacs receive blood from the fourth afferent branchial vessels. The respiratory membrane lining the air sac is thrown into folds and ridges and is composed of vascular areas known as respiratory islets that are the actual sites for the gaseous exchange. In case of the Clarius batrachus, the accessory respiratory organ that consists of suprabronchial chamber, airtrees, these are the airtrees, and fan and respiratory membrane. The suprabronchial chamber just lies above the gill, and it's divided into two cup-like compartments and is lined by highly vascular respiratory membrane, two beautiful rosette. These are the beautiful rosette that are known as airtrees on its sides that are supported by second and fourth branchial arches. The primary gill lamellae on each gill arch are fused to form a fan or gill plates that consist of also a vascular area. And then external gills. The external gills are highly vascular and filamentous outgrowths of the ectoderm covering the outer surface of the visceral arches. They are in direct contact with water. Therefore, the exchange of the gases takes place easily. The larvae of the Polypterus, they contain a single pair of external gills located in the region of the hyoids, and diploid larvae that possesses four pairs of external gills situated in the region of the visceral arches. This disappear completely in the adults of Lepidosiren but all retain as vestigial structure in case of Protopterus. To aid respiration, the embryos of the certain elasmobranchs that possesses long filamentous structure that protruding out from the spiracle. Now, what is the significance of the accessory respiratory organs? During development, the fifth gill arch does not develop gill lamellae, and its embryonic gill forms rudiments of the gill arch and aggregates to form a structure. That structure is known as gill mass. The air-breathing organs or accessory respiratory organs develop from gill mass. In some species, the gill arches other than the fifth arch also take part in the formation of accessory respiratory organs. The gill lamellae, which normally develop on gill arches for aquatic respiration, becomes modified to form the respiratory epithelium of the suprabronchial chamber or dendritic organs, and in some cases air sacs that also serves for aerial respiration. The accessory respiratory organ in Heteroponistes fossilis such as pneumatic sacs, whereas in case of the Clarius it is airtree, and that are the modifications of the gills. In this species, the swim bladder is either absent or greatly reduced, and during the Tertiary and the Quaternary period of the Cenozoic era, the oxygen level of the atmosphere and water was reduced due to depletion of oxygen in rivers and swamps. The gills were unable to cope the requirements of the body. Hence, the several teleostome species developed accessory respiratory organs to absorb oxygen from the air. And most of the fishes possessing air-breathing organs or accessory respiratory organs are capable of living in highly deoxygenated water. Of the swamps and muddy ponds infested with weeds, they have been observed to gulp in air from the surface and to pass it to the accessory respiratory organs. If prevented from reaching the surface, these fishes die due to deprived of oxygen. And accessory respiratory organs are capable of maintaining life of the fish in oxygen deficient water. Absorption of oxygen in the accessory respiratory organ is much greater than the excretion of carbon dioxide. Hence, most of the carbon dioxide is excreted in the gills, and the chief function of the accessory respiratory organ is the absorption of oxygen required for sustenance of life. These are the reference books and introduction to fishes, S.S. Khanna, Fish Biology, C.B.L. Srivastava, Fish and Fisheries, Pandey and Shukla, Analysis of Vertebrate Structure, Milton Hildebrand, and another book, Vertebrates, Comparative Anatomy, Functional Evolution, Ken V. Kadong. And some of the pictures in my presentation are collected from Google Image. Thank you, everybody.